Welcome to Biology Made Easy. In this lesson, we are discussing DNA replication part 2. And particularly, we are going to discuss the actions of DNA polymerase, RNA polymerase, DNA primase, primer, Okazaki fragments, ligase, lagging strand, discontinuous replication. All this is going to be discussed in this lesson. Well, DNA is a supercoiled structure. The enzyme topoisomerase relaxes the supercoiled nature and exposes the double helix nature of the DNA molecule. And so you have double helix. The enzyme helicase breaks the bonds between the nitrogenous bases and exposes the bases to be copied. In our last lesson, we discussed that the leading strand is copied in continuous replication. In this lesson, we are going to discuss how this strand is copied. Well, DNA polymerase moves in copying nucleotides in 5 to 3N. So when we look at the DNA molecule, this strand has 3' and it ends in the 5'. This strand, the black one, old DNA, the DNA polymerase needs to move to form a new strand for it. And the DNA polymerase will not move on the old 5 to 3 end because the DNA polymerase is moving to form a new strand that moves in 5 to 3. So in actual fact, the DNA polymerase cannot move on this because it's not the old strand is forming. It's supposed to form a new strand that has 5 to 3 ends. So the DNA polymerase will have to move from here, and the DNA polymerase cannot turn. So an enzyme called DNA primase is made. This DNA primase is an enzyme that catalyzes the formation of RNA polymerase. The RNA polymerase is able to make a short RNA segment, and that short RNA segment is called a primer. Now, let's have a quick discussion of how the RNA polymerase forms the short RNA segment. This is a DNA molecule that has opened up portion of it to be copied into RNA. Now, the RNA polymerase also moves only in the 5 to 3 direction to make new RNA. So this RNA polymerase, if this is the 5 to 3 strand and this is the 3'5 strand, the RNA polymerase will need to form a new one. So it will actually copy the 3 to 5 end so that it can make 5 to 3, 5 to 3, 5 to 3, 5 to 3, 5 to 3. Just like we explained for the DNA polymerase. So this RNA polymerase forms a short RNA segment from the DNA, copying the information from the DNA. And RNA polymerase can move on single strands. That is why when the DNA is opened, RNA polymerase can move on only one strand to form an RNA. But DNA polymerase cannot move on only one strand. So when these two strands are opened up and continuous replication is going on here, DNA polymerase cannot move on a single strand moving backwards. So the DNA primase makes a short RNA segment. And this short RNA segment moves in 5 to 3 direction this way. So we will use this diagram to explain. RNA polymerase can start moving from the back here. So it's going to form a short strand that has 5 to 3 ends. So the RNA polymerase at the back here and forms a short RNA segment 5 to 3 end, 5 to 3 end, 5 to 3 end, because it also moves in the 5 to 3 direction. And when it has formed a short RNA segment, the DNA polymerase will see the short segment moving in the 5 to 3 direction and continue the process for a short time. Then it will stop. So it forms a short fragment that is complementary to this portion. And these short fragments, they are pieced together by enzyme ligase, which is able to piece the short fragments together. And the short fragments that are made is called the Okazaki fragment. So this strand, that is how the discontinuous replication occurs. Because a primase will have to be formed, primer will have to be formed, and the primer is a short RNA segment. The polymerase will see its direction of movement that has continued, so it will also continue to form nucleotides. And when it forms the nucleotides to join the old one, the RNA portions will be broken off. And the strand is called the lagging strand because the nucleotides is not building up as fast as the leading strand where continuous replication is going on. Here, all these enzymes will have to go on and they have to move back. So it lasts. While this one has started folding up into the double helix, this one is now still fitting in new nucleotides. And so it's a lagging strand. This is what we call it a day. If you like the lesson, please give us a thumb, subscribe, and thank you very much. Bye-bye.